Good afternoon, everyone. Um, I'd like to thank the Brown Lab for hosting Maya Groner as our seminar speaker, but I have known Maya for a very long time, so I get the ominous duty of introducing her. So Maya earned her BA from Wesleyan College and her PhD from the University of Pittsburgh, where she worked with frog chytrid fungus. Fortunately for all of us, she moved into the realm of marine disease after that, first as a postdoc at Atlantic Veterinary College, primarily working on sea lice. But I met her through her interest in eelgrass, so she came out to Friday Harbor Labs and took the marine disease course. And we ended up working on one of the class papers together. We still collaborate on eelgrass, so we'll just imagine it's been over a decade and we have a current NSF project that we're about to wrap up. From there, Maya went to the Virginia Institute of Marine Sciences, where she worked on lobster and striped bass disease. And next, yes, there's a next. She was a research scientist at Prince William Sound Science Center in the USGS, where she worked on a lot of pathogens of native fish populations, including snow crab and herring. Now Maya runs the Quantitative Disease Ecology Lab at Bigelow Institute for Marine Sciences in Maine.
You all hear me? Yeah, mic's on. Okay, great. Well, I just have to thank Anya and Colleen and Serena for inviting me out. It's been a really special day getting to meet all of you, or many of you, and hear about your science and watch whales and just be in this beautiful environment. And I'm really excited to share the work my lab's been doing over the past several years. I've been at Bigelow for almost 4 years now, and this has been what's taken up most of our time, aside from the eelgrass work, which with Colleen, which I'm not going to talk about today. So I just wanted to mention Bigelow because it's a bit of a different model than an academic institute, and I thought some of you might be interested in learning about this type of model. We're a nonprofit organization where scientists bring in their funding, and we kind of each run our own little labs, but we don't teach, and we have quite a bit of freedom as far as what research we get to do. Most of the science is pretty global in scale, tends to be more microscopic. I'm one of the few more macroscopic scientists there. This is our facility, and you can see we have a brand new wing, education wing. This is actually just a computer-generated image of it, but it actually exists now, and we're hoping to offer more classes and bring people out. So if any of you are in Maine and want to stop by, we love having visitors. And I just did want to mention that I do work on eelgrass wasting disease, and I know there's a lot of eelgrass interest in this group, so I won't present on it today, but please feel free to reach out to me if you want to discuss this work. I love talking about it. So my lab is the Quantitative Marine Disease Ecology Lab. It's not just me. The folks that really helped with this work are Kirsten Johnston, who's a technician in the lab, Ren Yoshioka, who was a postdoc in the lab and was just promoted to a research scientist, and Melissa Rocker, who's a postdoc in the lab. And I really want to highlight them now because they did a lot of the heavy lifting of the work I'll be presenting. So in this group, so diseases are in our marine environments. They take a lot of different shapes and forms, and some can be quite problematic, and others are just sort of fascinating. And this is one of the more fascinating ones. This is a shrimp with a rhizocephalin parasite. So this is a type of barnacle that is actually eating the lipids out of the shrimp, and it produces this egg sac, which will release more of these rhizocephalin parasites, but the shrimp takes care of it as if it's its own young, its own eggs. So it's a really fascinating system, has very little impact on the shrimp population. We see this quite a bit in the Friday Harbor labs in the water around there, and the shrimp population is quite healthy. We can contrast that to other marine diseases, which you might not think of this as a marine disease, but highly pathogenic avian influenza is a disease that's impacting our marine environments. This is a case where scientists are looking at a dead seal that has been infected and died from this infection, and we're actually seeing mammal-to-mammal transmission of this virus. So this is a marine disease that not only has implications for marine health, but also for human health, potentially, that we need to keep an eye on. Just to sort of show the gamut of types of impacts we might see, I have to mention the sea star wasting disease. And you've probably all seen in the news the wonderful research that's happened recently to uncover the pathogen, the Vibrio pathogen that has been causing this. But this is a disease that has impacted over 20 species of stars and led to the federal listing of one species, the sunflower star. So I just wanted to share kind of a range of the types of diseases that we see and the types of impacts they have. And we really are just kind of at the tip of the iceberg in terms of understanding what these pathogens are, what's in our marine environments, what they're doing. It's a really exciting time to be a marine disease ecologist. In my lab, we think about a lot of questions. One is, what is causing disease? If we see a die-off, like these are Pacific razor clams at Olympic National Park in Washington State, we might wonder, is there a pathogen that's associated with this, or is there some other cause? In this case, we think it was associated with a pathogen, and we worked with scientists at USGS to develop an assay that was a chromogenic in situ hybridization assay. So it actually binds to the DNA of the bacteria that causes this gill disease, so we can confirm it. And we have a tool that you don't have to be a super well-trained histologist to be able to see these really bright spots in the gill tissue, that it's there. We also ask, has something changed to increase or decrease disease? In the Gulf of Maine, where I live, the obvious culprit is warming seawater. The Gulf of Maine is in the top 10% in terms of the rate that the seawater is warming. And so this is changing a lot of things like the hydrodynamics, but also the species compositions and thermal tolerance of many organisms is starting to push against that thermal tolerance. One example of an impact of temperature on diseases with this species, it's a striped bass that's impacted with mycobacteriosis. This is a bacteria that is it's similar to what causes tuberculosis, and it can cause these very slow developing chronic infections that eventually will kill a striped bass. And we worked with tagging data from the state of Maryland and Virginia, mark recapture data, and we were able to use that to calculate the survival rates of diseased and healthy fish. And we could calculate the survival of these severely diseased fish that looked like this, or maybe a little bit less awful than this one. But as the temperature increased in the Chesapeake Bay, you can see that the survival of these striped bass decreases to close to zero. And this is the central spawning ground for these fish, and we're actually seeing these temperatures now. So temperature is definitely a threat to many of our marine species, and one that we look at when we're thinking about changing disease rates. Another question we think about are the impacts of disease. Not all disease has impacts that we might be concerned about, but from a management or an ecology perspective, we might be particularly concerned if the disease is impacting a foundational or habitat-forming species like eelgrass, or a keystone predator like a sea star potentially, or a fishery like this snow crab fishery. And so for this talk, I'm going to mostly be focusing on two fish species, the snow crab and the lobster. And you can see from this NOAA graphic, both crab and lobster are some of the most lucrative organisms or species groups in the US. So both from an ecological but also from an economic perspective, we really need to pay attention when we're seeing disease in these groups. So some of the questions we asked with these two systems, with snow crab and with American lobster, is how is disease impacting some of our most valuable and iconic fisheries? Is there a role of warming seawater? And what can we learn by both looking backwards at historical data sets and maybe adding some new information from oceanographic models to that? And how does that help us predict and prepare for future disease threats? So getting into this idea of impacts a little more, I just want to mention how challenging it is to understand these and track these diseases in marine systems. Right? Diseases can be very hard to observe, particularly if there's mortality. Dead organisms don't last very long in our oceans and may be swept away or moved by currents. We have very complex mechanisms for dispersal of pathogens. They can survive in seawater, many of them, and disperse long distances. We often have huge knowledge gaps in both pathogen and host life cycles that can make it challenging to optimize our sampling. And sometimes we have challenges with feasibility of lab and field studies, particularly an organism like the snow crab, which can be quite expensive to even get out into the Bering Sea to sample. You can imagine that just going out and collecting some is not really an option unless you piggyback on some research that might already be happening. So how do we get around this? What I like to do is really work in collaboration with state and federal agencies because they're often collecting a lot of data associated with fisheries populations. They're often out in the fields. They've got their boots on the ground or in the water, observing potential changes. And we can use their data and feed our own information from disease surveillance or experimental work, and perhaps oceanographic models that might tell us about sea conditions when these organisms were sampled. They actually allow us to go back in time to hindcasted conditions. And we can feed all this into mathematical models and come up with some predictions about what might happen in the future. So I'll demonstrate this in some of the ways we go about this with the two case studies. So the first is with epizootic shell disease in American lobster. This is a severely diseased individual here, but what you can see is that its exoskeleton is not okay. It is infected with a dysbiotic suite of bacteria that are actually consuming the chitin, the material that forms the exoskeleton, and that bacterial suite changes over the course of the infection. We know from both mark recapture studies and experiments that severe cases like this are lethal. Less severe cases might not be lethal, but these lobster will have very little value in the fisheries market. They can't go on the dinner plate, and so they go straight to a cannery at a very low price. Direct transmission, we haven't really been able to demonstrate that, but we can cause lobster to break with disease just by increasing temperature or causing other stressful situations. So what we think is happening is that the bacteria that cause this infection are already on the exoskeleton, and the balance of bacteria changes in stressful situations and at warmer temperature. Histologically, this is a healthy lobster, and you can see that this is the exoskeleton, or exocuticle and endocuticle, and then just the tissue underneath. It's very even, organized. We can contrast that with the histology of an infected lobster, and you can see this is a very characteristic pillar-like structure that we see in the degraded exoskeleton. And these purple areas indicate bacterial infections that are occurring on the outside of the shell. Fortunately, when this disease first appeared in the late 1990s in Long Island Sound, scientists at the Millstone Power Plant started doing mark recapture studies and collecting data on the prevalence of disease in the lobster population. So that every year they would go out from May through October and quantify the proportion of animals that were diseased. And so you can see in May, that's sort of what these check marks are, the sampling period, and then there's a blank spot in the winter when they weren't sampling. So each year they had somewhat moderate levels and it declined and then it increased again. Any guesses as to what's causing that pattern? Why isn't disease just increasing? This is from May through October for each of these check marks. Lobsters are molting. So they molt out of their diseased exoskeleton. They're healthy for a while, but it's this warm summer, and so the disease increases very rapidly after they molt. You can see some years it's quite high. These tend to be really warm years, and other years the maximum prevalence in October is somewhat lower. So we know molting and temperature are quite important for this disease. And I'm just going to go quickly over some results. When I was a postdoc, I analyzed this data and was able to show that there was kind of this phenological mismatch where the warming temperature was changing the timing of the molt cycle in a way that led to more severe disease and more prevalence of this epizootic shell disease. So this is just a diagram of what we thought was happening from these data. So if we had a population of lobster and it was warm in the winter, that would lead to early molting. Warmer temperatures tend to cause molting. And so you would have a healthy population in the spring, a relatively healthy population. But that would cause, as a result, you get this long summer intermolt period, which is when the disease is most likely to be transmitted or to occur. We don't think it's transmitted. And that transmission that occurrence could be exacerbated if it was a particularly warm summer. So you get this increased prevalence of disease. And then we found that actually that the disease mortality tended to occur most in winter. So we have these sort of complex interactions between temperature, disease progression, and molting. But this was in southern New England. And I live in the Gulf of Maine, which is the epicenter for the lobster fishery. And so I wanted to know if epizootic shell disease is going to impact this fishery. So with collaborators from the state of Maine and Massachusetts, we got a Sea Grant award from the American Lobster Initiative that allowed us to do experiments and modeling to predict the future prevalence of epizootic shell disease in the Gulf of Maine population. And I have to point out Melissa here, she really led the majority of this work. So these are the two stocks that we have in the US, the southern New England and the Gulf of Maine stock. And if we look at the southern New England population of lobster, you can see that the lobster peaked in the late 90s and the population crashed. The fishery crashed, it has not recovered. And that coincided with a lot of warming as well as epizootic shell disease. We don't think that disease alone caused this crash, but it certainly contributed. We look at Gulf of Maine, and I have to apologize, the stock assessment isn't released that often, so we don't have data past 2018 until later this year. But the abundance was increasing. It's actually kind of plateauing now. And that is thought to be because for two reasons. One is that the water is warming in the Gulf of Maine, so we're kind of at this thermal optimum for these lobster, and also that fishing is increased, and then the lobster fishery is incredibly inefficient. So, which means that people are putting food out in these pots. Lobsters are going in and out, eating food, and so we're almost ranching them, right? So we're feeding them, and so the population grows. So we kind of have these two reasons. We've got this very lucrative and abundant lobster fishery in the Gulf of Maine. But knowing what happened in southern New England, there are a lot of concerns about the future of this fishery. Maine state scientists have been tracking epizootic shell disease in this population. So these are all of the Maine state fishing areas. And you can see that the prevalence has increased over time, particularly in these purple areas. So this is kind of near Portland, Casco Bay, whereas down east in the Gulf of Maine, we tend to see much lower prevalence. The water tends to be colder. This is really different from what we see in Long Island Sound, right? The proportion of individuals with disease is maybe 1.5% at most. I have to point out that they're only, in these surveys, they only measured disease in lobsters that had at least 10% of their carapace. So it's a slightly different estimate that we're comparing, but it's a much slower takeoff of this disease. So we want to understand why. Is there something about this population that makes them more resilient? Is it just that the temperatures are different, or do they have a different molting pattern? So ultimately, what we wanted to be able to do was to create an individual-based model where we could have lobster that we know start as disease-free, and that over time, they go from these different severity stages where they have different proportions of their exoskeleton with lesions. And at each stage, there's a probability of molting and a probability of mortality. And we can make all of these transitions temperature dependent. We need data to do this. And so Melissa ran an experiment with the goal of understanding these epidemiological rates at different temperatures. So what she did was collect lobster from Maine, Massachusetts, and Connecticut. So both of the Massachusetts and Connecticut lobster were from the southern New England population that's already experienced the crash, and the Maine are the healthier population. And we had these lobsters at 12 and 20 degrees Celsius. You can see the experiment here. The lobster were housed individually, and we kept track of their molting and their disease status over time. And from these data, we were able to estimate epidemiological rates. So here we have both the Maine, or three populations, Maine, Massachusetts, and Connecticut, over the 140 days of the experiment. And we can look at the coverage of disease, the amount of the exoskeleton that's diseased at these different temperatures and in these different populations. And Maine stands out right away because we see much higher rates of disease progression, and it's occurring both at warm and at these cooler temperatures, where in comparison, Massachusetts and Connecticut are seeing very little disease progression. So this doesn't look great for Maine, but when we look at molting, we see a different pattern. So here we have Maine, Massachusetts, and Connecticut, and then, this is cold up here and warm up here. And then these colors represent the amount of the exoskeleton that is covered with epizootic shell disease. So the most infected, most diseased lobster we saw had about 50% of their shell had lesions on them. And so we were able to model the probability of molting as a function of these different levels of disease. And you can see right away, again, that these Maine lobster are molting much more at both cold and warm temperatures. And across all of the populations, the more diseased an animal is, the more likely it is. So we thought this was pretty interesting. Not only is disease progressing more rapidly, but they're also able to kind of escape disease by molting a little bit better. So predicting the impact, kind of the net response of these two factors is a little hard to do. So we went back to our individual-based model, and we added these molting probabilities and the incidence and progression rates that we had for disease from our model. And then we made these temperature-dependent rates. We also added mortality values from the stock assessment models, and we created our individual-based model. So really, Melissa did this. So let's see if I can show you. Yeah. So the model's running. Right now, she's just got a model where she has a population of lobster at a low constant temperature. I think she has it at 12 degrees. And she's looking at, this can't be 12 degrees because there's a lot of mortality. So I'm not quite sure what the conditions she has here are, but you can see she can track over time the proportion of the population that's disease-free or has different levels of severity of epizootic shell disease. So right now, these are just kind of made-up numbers that are in here. I think she actually has a better model that's a little more realistic that we're currently validating against the historical data that has been collected by the state of Maine. So we've done this model in a particular way to have this like visual representation because we want to be able to share this with managers and also lobstermen. So to communicate what we've predicted and not just share graphs and numbers, and also have them maybe tell us if this matches what they're observing when they're out in the water. So for this section of the talk, I'm not going to go into too much more detail about what we're doing, but the conclusions are that we're seeing these Gulf of Maine lobster develop disease more rapidly than the southern New England lobster, at least in this experiment. And I have to add the caveat that we collected these lobster from the wild, so they're kind of acclimated to different conditions initially. These are 4- and 5-year-old lobsters, so it would slow us down a lot if we raised them in common conditions in the lab to get them to this stage. But sort of the hope for the Gulf of Maine lobster are that they are more likely to molt in the summer out of this disease. So that might be the saving grace as the water warms up. So we're showing that we were able to show that both disease progression and incidence increased with temperature. And we're in the process of using that 20-year data set to kind of validate our experimental results against the field data. And then finally, we'll use this individual-based model to identify model sensitivities. So are there different factors in the model, like assumptions about how often the lobsters are molting or what triggers the molting, that really change the outcome that might help us understand future research directions. We might be able to identify climate thresholds, certain levels of warming that could lead to more sort of rapid, severe outbreaks. We want to use this as a communication tool, and we're hoping that these results from this model will inform the 2030 stock assessment for this fishery. So I can take any questions about this before I kind of take us over to the Bering Sea for our next case. Or we can wait till the end. Oh, yes. Oh, repeat the question. So the question was, did we inoculate the lobster with the pathogen? And because this is a dysbiosis, it's not like one pathogen, we don't. We've actually found that the lobster, they actually have the bacteria on their shell already, it just, as you increase the temperature, they break with this disease. So one thing we did do was we didn't completely sterilize the seawater as it was coming in because we wanted the natural bacteria in the water to be able to colonize the shell. We thought that would be a little bit more realistic. Any other questions? Sorry.
All right, so this one's from Zoom. So Ted Groschultz asks, does this disease influence reproduction success or timing?
Yeah, so I should have mentioned this. So the disease, the longer the intramolt period is, the more the disease can progress. And in female lobster, adult female lobster, producing eggs is incredibly energy intensive. So they actually molt every 2 years, as opposed to adult males, which molt every year. So we tend to see much higher disease prevalence in adult female lobster. Studies so far don't show an impact on fecundity, but the survival of those females is most likely to be impacted before the males. One thing we do find, though, these lobster, they mature to this like yearly molt right at the time they recruit into the fishery. So if there was no fishery and these lobster could live as long as they wanted or were able, we'd probably see a truncated survival, but the survival is truncated even more by the fishing. So some of the impacts that we would see in an unfished population are sort of a moot point because very few lobster make it out of the, almost every lobster is fished when it recruits.
Yeah, I thought it was really interesting that the Maine lobster are molting more. And as you pointed out, you were using field collected lobsters. So I was wondering if you had to speculate, do you think that's some genetic divergence between these two stocks, or is that some plastic environmental effect of growing up in the Gulf of Maine versus southern New England?
I think that's a really good question that we can't answer from these data. There has been some genetic work on the stocks, and it's somewhat dated, and there wasn't much variation that was detected between the southern New England and Gulf of Maine stocks. But I don't know if and I'm not a population geneticist either, but I don't know that if with more modern techniques, if there might be some more genetic variation detected. I do think these southern New England lobster have been exposed to pretty warm temperatures. There's potential that they were primed for future. Yeah, it's a good question. All right, so moving on, the next portion of the talk, I want to take you to the Bering Sea, and just to orient you, this is the eastern Bering Sea. This is on Alaska, or where the town of Dutch Harbor is, where most research cruises go out to study this area. The snow crab fishery takes place along this shelf here. It's about 200 fathoms deep, I believe, and that's where the snow crab thrive. The Bering Sea has historically been a sea ice dominated system. That's really critical for the productivity of this system. Historically, the plankton, they are growing right under the ice. And so as that ice has been lost in recent years, it's actually causing some pretty major regime shifts in terms of what organisms are here and the productivity. So how fast is this area changing? Here's some NOAA data that show average bottom temperatures in 2010, 2017, and 2019. If you're a snow crab, you like water that's 2 degrees and lower. So kind of this purple area, which is called the cold pool. So in 2010, you had a lot of great habitat. There was a heat wave in 2017, it shrunk by half, and then by 2019, it was about a fifth of what you saw in 2010. So in this year, I almost said lobster, the snow crab were pretty much only found in this area at very high densities. They were really smushed into a small space and competing for a lot of resources. The snow crab fishery collapsed, and it collapsed 2021, 2022. Research cruises were canceled because of COVID, so we don't know the exact timing of this, but it's an estimated 10 billion lobster. No lobster were lost from this fishery. Snow crab, 10 billion snow crab were lost from this fishery. They failed to recruit. So they were seen as juveniles, and then several years later, when they were expected to be at a fishable size, they were not there. So this is, if you live in the Aleutian Islands in Alaska, this is a major source of funding for taxes, for schools, for roads. It's a huge loss, not just for the fishery, but just kind of all the ways economically this money trickles through that region. During this time period, a new disease or syndrome was noted in these crab, which was called black eye syndrome. So these crab, their eye stalks were melanizing under the cornea and eventually eroding into these stumps. This disease or syndrome, which we'll call BES for black eye syndrome, was first observed in 2017. So that kind of coincided with the beginning of that heat wave. Histology was not able to, initial work was not able to find a causative agent, and it was unclear if this was impacting the fishery. So to address this question, we took a very similar approach to our lobster research, where we assembled a team of scientists that were federal, both from NOAA and from US Geological Survey. We got a crustacean disease expert, Hamish Small from BIMS, and then we got state scientists from the Alaska Department of Fish and Game, who really had, they knew about the fishery, they could tell us their observations, and we could help about what this might be doing to the fishery. This work was funded by the North Pacific Research Board. So here's our research team. Just wanted to point out that we are really diverse in terms of the different agencies and academia that's represented. And I think this is key to having really successful collaborations within disease ecology. So here's some histology. This is the eye stalk of a healthy snow crab eye. These are the ommatidia, and this is the cornea. You look at a diseased eye, you can see the cornea is completely damaged, the ommatidia, there's some, but many of them are missing. There's melanization here. This crab could not see, which they actually do rely on vision, even though it's fairly dark in the areas that they inhabit. But of maybe equal concern, the eye itself has a gland that releases molting hormones and hormones associated with sexual maturity. So we were concerned that those might be affected as well. So in this project, we ran both experiments and did some modeling to try to understand the impact of this disease on the fishery. I'm really going to focus on the modeling just for the sake of time, and just a little bit of the experimental results. So here what we wanted to do was model correlations between environmental and demographic conditions that are associated with black eye syndrome. And similar to the lobster, we were extremely fortunate in having a huge data set that was collected by the state. I think I forgot to mention we had over 1.5 million lobster observations over 20 plus years for the ESD data that I showed you. And in this case, we had over 700,000 observations of crab that were taken by crab observers. So these are people that are sent out on the fishing boats to monitor the catch. And they were instructed as how to detect the visual signs of this disease. So of this 700, so they were able to get these data. And I put this picture here just to point out how precious each of these data points is, because they are out in the Bering Sea in the winter, and it's not easy conditions. We also had data from this Bering 10K oceanographic model that not only models the surface temperature, but they now have the bottom temperature conditions where these crab are, as well as modeled pH and dissolved oxygen. So we could take these data and start to look for any spatial temporal correlations and correlations with these environmental data. And this work was led by Ren Yoshioka, the other postdoc in my lab. So he gathered these data sets, the environmental data sets and the observer data sets. He spent quite a bit of time cleaning the data, making sure there were no typos and it all made sense. And then he took out a number of factors, such as whether they had BES, how big they were, if they had new or old shells, so how recently they had molted, and the fishing effort, which is sort of a measure, sort of a proxy for how dense the crab might be. From the environmental data, we could look at the bottom temperatures, pH, and dissolved oxygen. And then he could use a mesh to spatially and temporally match these data sets. So create these grid areas and see where the data overlapped. So here's his mesh. This is a very complicated process that I'm kind of summarizing quickly. And then he used a mixed effects logistic regression that accounted for this spatial temporal random effect. So a lot of steps to try to tease out these environmental and demographic drivers. And then he was able to interpret his results. So here are some of the key results. So black eye syndrome and environmental conditions varied over space and time, but generally the prevalence was quite low. Of those over 700,000 observations, we had less than like 40,000 observations of black eye syndrome, but there were some sites that were incredibly hot and many sites that were very low. I think the hottest site maybe had 65% of crab infected. You can see these prevalences are less than 1% in many cases. So generally quite low with some very, very hot spots. And I also have to point out, this is only about half the data because of confidentiality rules. We couldn't show data points unless there were at least 3 boats fishing in an area. So it's not a full coverage of what was actually going on. There were strong correlations with some demographic variables. So the bigger the crab was, the more likely it was to have black eye syndrome, and the older the shell was. So these crabs terminally molt, so once they reach maturity, they don't molt again, and they just get more and more barnacles and anemones, and they actually get this little leech that grows on them. So we could really tell what these old crab were because of all of the fouling on them. So those crab were more likely to have black eye syndrome. And then there was kind of maybe this marginal correlation with density. But the biggest driver we found was temperature. So there was at sort of these optimal conditions, we saw very little black eye syndrome, and that increased as the sea bottom temperature warmed. Interestingly enough, however, these data suggest that we have these kind of crusty old shell lobster sitting in, I think of them as sitting in a hot tub, but it's really 3 degrees Celsius, 4 degrees Celsius, that are getting these black eye syndrome. But we ran an experiment, and we could not get these temperature effects to show up in the experiment. So we had these crab in Kodiak, Alaska, similar to the lobster experiment. We could not demonstrate transmission of black eye, so we co-housed these also due to just limitations of the lab spaces. And we had them in replicated tanks of either 3 degrees or 6 degrees Celsius. And we had 96% survival, regardless of temperature, regardless of the black eye syndrome, the status of the eyes. We ran this for 6 months. We were able to estimate the incidence, so rate of new cases and the recovery. And what you can see is they're generally getting disease at a faster rate than they're recovering from it, but they're kind of going back and forth. They are recovering unless they get severe disease, at which point their eye stalk is eroding to a stump, and it's very difficult, if not impossible, to recover from. So we don't know what that temperature effect was driven by, but we don't think it was a direct effect of temperature, but maybe it was by something else in the environment in our field data. So this suggests that black eye syndrome is not really impacting this fishery. It probably had very little to do with the snow crab collapse, but we think maybe it's more of an indicator of stressful conditions, that as the temperatures are warming, maybe you start to see that showing up and sort of something the fishery can take into account as a warning sign. We did, however, find that these moderately and severely diseased I'm gonna keep calling them lobster, crab, they had very few lipids. So it's not that it's totally harmless. We think it's something's happening to their energy storage or energy use when they're infected. Okay, so we're waiting on data on the microbiomes associated with this disease and some transcriptomic data that might maybe help us figure out what the causative agent is. It's been kind of held up in the federal system at the moment, but we're hoping to get it soon. Okay, so just some takeaways. We used some data streams, like the modeling, the oceanographic modeling, and collaborations with Alaska Fish and Game and the observer data to make inference that was otherwise impossible to make. We could not run studies independently out in the Bering Sea on these crab. So it was a really successful collaboration in pretty rapid response, as this disease was only detected in 2017. We did find that temperature was correlated with this disease. We don't really understand how. We don't think it's a cause of mortality. But I think what we're really proud of in this group is that we thought we came up with a pretty good framework for responding to a disease that was recently detected, unknown etiology, and just an approach to work with the fisheries managers to try to understand impact and hopefully etiology or causative agent at a relatively fast time scale, we're hoping. So just as a teaser, we're currently working with most of the same collaborators, so folks at NOAA, Virginia Institute of Marine Science, and Ren, the postdoc in my lab. We're working to look at another disease in this fishery, which is called bitter crab disease. And it gives these crab this kind of cooked appearance. So these ones on the left have bitter crab disease, and the ones on the right are healthy. And they had this almost cooked appearance because their hemolymph is just teeming with a parasitic dinoflagellate. And so it kind of makes it white and milky. And so we know this is a lethal disease, and we're concerned about the impact this may have on the fishery, and we're beginning to study these data that Alaska Fish and Game have collected on this disease. Hamish Small has developed a new diagnostic tool to detect this pathogen with a digital PCR, and we're beginning to evaluate patterns of disease across sites. So this is just kind of a teaser of data, but this is from the site we had the most infection at, and this is prevalence, so percent of individuals that are diseased. And you can see we use 3 different methods to look at disease. So historically, they've just looked at visual diagnostics, so just do they have this cooked appearance? They found about 50% in 2018. And then with this digital PCR, we separated it into 2 groups, all of the positives that we had, and then the ones that were really high positives. And you can see there's actually a really good match between the visual and the high levels of infection. But 50% of the surveyed animals had this disease, mostly small crab that were surveyed in 2018. It dropped a bit in 2019. This is when the population crashed, and now we see much lower levels of the disease. So we're trying to piece apart what might be driving that using similar approach. All right. So to summarize from these examples, what have we learned? Climate change is definitely affecting our biggest fisheries, and they're happening in areas, these fisheries are occurring in areas where water and, in the case of the Bering, ice conditions are changing, and infectious diseases are a concern, but not always the cause of the mortality. I think, hopefully I've demonstrated that I think we have a lot more impact doing this research when we work with managers, and we collect data with them or leverage their collection efforts and the communication routes that they have with the various members of the fishing industry. We need these multidisciplinary teams. We need the experts in histology and molecular techniques and modeling and the fisheries themselves. And I'm hoping these models will allow us to learn from our past and start to prepare for the changing future. Running out of time, but I just have a couple more slides because I think it's important to talk about what's next. What happens when you detect these diseases? Can you manage them? What's next if you know there's a disease in your system that you're studying? So this is from a paper from 2016, where we looked at kind of just had this thought experiment where we have time and we have disease incidence and you have this characteristic epidemic curve. And if you're not doing any surveillance, you don't really notice a disease outbreak until probably after you've had your epidemic peak. And then these impacts, which are in these dotted lines, can be quite high. When we start adding surveillance methods, maybe you detect the disease a little earlier and you can mitigate some of the downstream impacts. And if we can get to this point where we're forecasting disease, maybe we have more opportunity for mitigation. And so that's what we're hoping to get to eventually with the lobster fishery. So once we know that's happening, what else can we do? Well, some of the work that Colleen's been working on, that I've collaborated on a little bit, shows that at least in some cases in coastal systems, you can actually use other organisms to help filter pathogens out of the environment. So both seagrasses and oysters have been shown to reduce abundance of certain pathogens. So just habitat restoration in some cases might make some systems healthier. In the case of fisheries, there's a lot of evidence that disease can impact fisheries, but also that fishing, I think, can impact disease. So when you fish, you're culling individuals out of the environment. And this is a figure by Chelsea Wood that I really like because she shows all the ways the fishery can impact the disease. It can target the host, it can just target infected hosts, it can target uninfected hosts, and then it can target other individuals in the community that that fishery exists within, like predators, competitors, and prey. And so with all of these different types of fishing, some of them lead to decreases in parasites and some lead to increases in parasites. So when we're fishing, we're changing the abundance of parasites or pathogens in systems. So maybe we can do that a little more deliberately. So there's certain ways I think we can work with stock assessment scientists to incorporate disease into their management plans. Currently, we've just done this with sort of a case study with a hypothetical data set with a viral infection, just demonstrating that this can be possible to actually put a disease model into a stock assessment model. But I think there's a lot of room for creative approaches. And they might not be as quantitative as this. They could be just changing thresholds or using disease as a indicator of stress. We can also just increase awareness of marine diseases. I think it's not something that is on a lot of people's radars. And if there is a management action that needs to be taken, you sort of need trust within that community for it to be accepted. So this can be done with talks to the public or to fishermen or managers. This is a picture of a disease course that I taught, and I taught in the past with Colleen to train future scientists to work on this. And this is a game that was developed by Ren, the postdoc in my lab, called Crabdemics, which was designed to teach high school students about diseases. And so they're just more literate in this kind of terminology and understanding potential management approaches. So I think there's a lot of potentially fun things to do to increase literacy in this area. So that is it, and I would love to take your questions. Thank you. Yeah. Oh, okay. I did put my email address at the bottom if you have a question later that you want to ask.
Hi, great talk, thank you. I was wondering, do you think it's possible that crabs infected with black eye syndrome might exhibit higher rates of mortality in the field because they can't see, whereas if you're raising them in captivity, like maybe that's not as important?
Yes, yes. So yeah, there's actually a study, and I'm not quite sure of the details, because that was done that basically showed that vision was quite important for snow crab, and I think they were actually able to model what a snow crab is able to see. And I know that some fishermen will actually put lights on their traps to track the crab, and it works. So they are responding to visual cues. I didn't mention that we did an experiment where we wanted to look at chemosensory behaviors to see if maybe they were compensating for damaged or loss of eyesight, just with looking at antenna flicks in response to a stimulus, and we did not see an effect there. But certainly predators as well, maybe they're not seeing predators. We don't tend to see the really, the crab that have the stubby, eroded eye stalks in the field, which makes me think they're probably not surviving.
Along those same lines, thinking about the experiment with the increase in temperature, of course, that will increase their metabolism and potentially increase the amount of food that they require. But if they are in the lab, they're probably being fed all the time, whereas
Yep.
if they're in the field, that increase in temperature can impact, and then you'll have two stressors, right? Low lipids and
Yeah.
also, so in the lab, are you able to starve them and see if you, or give them low amounts of food and see the interplay between food and temperature?
It's a really good question. So there are some studies that have looked at metabolic rates of snow crab in response to increased temperature, and I think it's like a 2-degree increase can double their metabolic demand. Everything is happening very slowly at 2 degrees Celsius, so a small increase certainly increases their demand. And the hypothesis for the snow crab collapse is, the working hypothesis is that these crab were forced into a very small area that had a temperature optimum, and they were competing for food, and they starved. So it was both that increased metabolism and the increased competition that was facilitated by the temperature. There is a lab that has, not looking at black eye syndrome, but just looking at metabolic rates and survival of crab. I think they've been looking at that question of starving crab at different temperatures. And again, because everything happens really slowly, I believe the last I heard was that experiment had been running for 6 months, and the crab were just resilient. So it'd be interesting to find out what the outcome of that is. In our experiment, we did the crab were fed more than they could eat. So there was no limitation of food. But even with that, we found that the lipid content of the crab was in the hepatopancreas, which is sort of the lipid storing organ. It was close to 0 in the severely diseased crab, regardless of temperature. So even when food is not limiting, it seems like this disease is having an impact, but at least on the time scale of this experiment, we didn't see that it affected survival. And I guess with the very low prevalence, I don't think it could impact the population that much. Big question.
Thinking about the fact that all these are fishery dependent data, and then also thinking about that host and parasite interaction flow chart that you had, have there been any studies on whether or not these diseases or parasites affect the meat quality in the different species from striped bass onward?
Yeah, yes. So bitter crab disease in particular, as you might guess from the name, gives the crab, I've heard it's like almost like an aspirin-like flavor. So they're not marketable again. Yeah, so that affects the quality of the meat. And I think with the other diseases, it's more about appearances and just people aren't comfortable eating an animal with lesions on it, even if it's not going to infect them. So yeah, there's an impact on the economics.
All right, I know we had a Zoom question, but does anyone else in the audience have any more? Oh, okay, thanks.
So you mentioned briefly that crabs have organs associated with their eyes that are involved in the molting hormone. So is there any evidence yet whether crabs that are infected with black eye syndrome are or are not able to molt?
That's a great question. That was the question we came into this project really interested in, and we thought, okay, we can like bring them into the lab and see if they molt. No crab molted in the lab, but the crab that we caught, so we caught them, we went out on the NOAA trawl survey and got these crab to bring back to the lab and tried to get, similar sizes with some with black eye and some without. And they were almost all terminally molted males. So it didn't really let us look at that question, but I know with this crab you can, if you remove an eye, experimentally, it can trigger a molt. But that organ it's really deep at the base of the eye stalk. And so it's unclear to me if this disease is impacting the organ. We're not really seeing that histologically, but yeah, it's an open question.
All right, maybe we can just finish off with a Zoom question I did not get to before. And this is about the lobsters. All right, so Peter Alpert asks, what might cause the increase in molting probability with intensity of disease?
Yes, so there's two theories on this. So one is that a lobster has more disease because there's been a longer period of time since it last molted. So it's not that there's actually an increase in molting probability, it's just that you're more likely to be diseased right before you molt. In the experiment we did, we staged every lobster. So you can look at their pleopods and stage like how, if they're an intramolt, or if they recently molted, or if they're about to molt. Our lobsters were all intramolt lobsters, except for, maybe there were like 5 that were slightly closer to molting. So I think it was a C4 and a D0, if that means anything to folks here. But we didn't see that molt stage had a huge effect on the probability of molting with disease. So the other thought is that the disease itself triggers an increase in the release of a molting hormone called ecdysone. So it's an adaptive response to get rid of the disease that probably comes at a very high energetic cost because molting is quite a bit of energy.
Awesome. One very, very last question. Can I purchase that board game, the Crabdemics one? Is it available online?
Ren has been asked that question a lot. At the moment, there's like 16 sets in existence. But yeah, I'm hoping, I think he's realizing there might be a market.
Okay. Awesome. Well, everyone please join me in thanking Maya.